of my scientific friends, at least on the screen. And I really hope to catch up in person, hopefully in 2021. So before I introduce Fabiana, just a few words on the structure of the session. Fabiana will present uninterrupted for 30-some minutes. Please submit your questions, comments using the chat option of Google Talk. I will try to relay those to Fabiana after she ends her presentation. And in the conclusion of the group discussion, Fabiana kindly agreed to stay connected for some extra minutes, so you can also talk to her directly. So now a few words on Fabiana's research. As many of you know, the molecular composition of the mitochondrial calcium uniporter resisted efforts to solve it for many decades. And this gap hindered the study of the relevance of mitochondrial calcium homeostasis in cell function and dysfunction. Fabiana brought a new systematic approach to this problem, which builds on analysis of genome-wide data sets, evolutionary clues, comparative physiology. She started to build her toolkit with Lars Steinmetz at EMBL and applied it to the uniporter in Vamsi Muta's lab with inclusion of organelle proteomics. In 2010, Fabiana's efforts led to the discovery of the calcium sensing gatekeeper of the uniporter, MICU1, and in 2011 to the discovery of the pore-forming unit of the uniporter, MCU. Notably, MCU was co-discovered by Diego De Stefani and Rosario Rizzuto. In my opinion, these findings were among the most impactful discoveries of molecular mechanisms in the 2010s. After returning to Europe, Fabiana made a stop in Barcelona and then set up her group in Munich, Germany. Applying her distinctive strength in bioinformatics, she continues her efforts on mitochondrial calcium transport, and these efforts have given rise to several quite important and interesting papers recently, and she will give an update on these studies today. So, Fabiana, please take over here.
I'm most grateful for the very kind introduction, Yuri, and I'm very happy, of course, to share some of our progress with all of you. And thanks also to Martin and Kadia's invitation. This is a great platform for exchanging science. So I'm quite delighted to be able to speak today. Of course, it's kind of a good and bad to have a ghost audience, but at least some of you could have the freedom to take a nap here and there if I may get too boring. So as Yuri already introduced, most of the research in my lab focuses on studying the mitochondrial organelle. And to this goal, we take a systematic and integrative approach, which combine, from one side, computational large-scale experimental strategy with traditional biochemical, genetic, and physiological analysis of several mitochondrial functions in different model organisms, with the overall aim to systematically predict, characterize novel mitochondrial proteins and pathways, and to be able to modulate and target eventually them in specifically our interest would be in neurometabolic diseases such as diabetes and Alzheimer's. So we were used to think of mitochondria simply as the cellular power plants catabolizing nutrients to produce ATP. In fact, only 1% of the mitochondrial system is actually dedicated to such a function. While we know now that the organelle engages in decoding many cellular inputs, thus controlling a variety of processes within the cell, from the biosynthesis and catabolisms of macromolecules to ion homeostasis and even to immune responses. So how can the organelle actually sense and process so many inputs? Well, first of all, they are dynamic. They form tubular networks that constantly remodel throughout the cells. And in specific cell type, like you see in the middle for neurons, they can even travel long distances, just like trucks on a highway, towards intracellular pit stops where they provide energy and metabolites. And at the same time, like us, they are, like we do on social media, they are highly integrated into cellular network by exchanging information constantly with other organelles through physical or functional interconnections, like you see depicted here with the ER. And such a pleiotropic role is actually reflected by the complexity and the heterogeneity of the mitochondrial proteome. At least in mammals, this is estimated to consist of about 1,500 components. They vary a lot in both the genetic and evolutionary origin, but also 15% of the mammalian mitochondrial proteome has been estimated to be tissue-specific, and even differ among cell type of the same tissue, as we recently showed for the cerebellum. So this allows the organelle to meet the unique metabolic and physiological requirements of each tissue and different organs in different organisms. And therefore, it shouldn't come as a surprise, of course, that the dysfunction of many mitochondrial genes, more than 300, have been already associated to over 190 disease phenotypes. But this is just an underestimation if we consider that roughly 40% of the mammalian mitochondrial proteome still remain completely uncharacterized. One central question that my lab is currently focused on is actually the reconstruction of signaling, of calcium-dependent signaling networks in mitochondria, which allow the organelle to sense, to decode, and to respond to the cellular needs I mentioned before. And one such a universal mechanism that it's pretty much conserved in mitochondria from almost all eukaryotic cells is the use of calcium ion as a messenger. So when the cell gets stimulated, or receive a stimulus, the cell increases the cytosolic level of calcium, which is then actively imported into mitochondria. And here in the mitochondrial matrix, by signaling cascades that still remain mostly uncharted, there is an increase of mitochondrial calcium concentration, which is then coupled to buffering of cytoplasmic calcium kinetics, waves, like you see in this case for a Xenopus oocyte after fertilization, shaping the dynamics and the distribution of these waves. It is also coupled to the production of energy to increase the energy production in order to sustain the stimulated cells. And when the level is too high, can actually act as a checkpoint to activate several pathways of cell death. And several diseases have been already associated to dysfunction of mitochondrial calcium homeostasis, actually in both directions, linked to either a decrease or an increase of mitochondrial calcium uptake. Despite the recognized importance of the mitochondrial calcium signaling in physiology and pathophysiology, like Yuri mentioned, the molecular players that mediate this process have remained, and let's say still remain, largely unknown. On one side, how does calcium enter the mitochondria? Then how does calcium is coupled to mitochondrial function, and how mitochondrial can actually transduce the calcium signal into specific output that affect the life, the fate, and the development of the cells. And as Yuri pointed out, we now in my lab, we take a systematic approach using both computational and experimental strategies. And today, I just want to focus on giving you a couple of examples of how a this type of approach can actually catalyze the understanding of mitochondrial calcium signaling. And the first example, so the first part of my talk, I will show you how the evolutionary genomics approach can be used to discover and characterize both the genetic determinants of mitochondrial signaling, mitochondrial calcium signaling, so to go from function to protein, and also how it allows us to go from protein identified to characterize their mechanistic role. The first example I take, the following example, dates back to my time as a postdoc in the lab of Vamsi Muta. So at that time, I was looking for a strategy that would allow me to identify de novo the founding members of this so-called mitochondrial calcium uniporter, which was known to be a calcium selective channel responsible for the uptake of calcium in mitochondria. And I was quite fascinated to see that studies already back in the 60s and the 70s demonstrated that not all eukaryotic mitochondria were able to take up calcium, as you see here. In fact, fungal species, including the widely used model organisms, Saccharomyces cerevisiae, the budding yeast, were not. So this led me to make a simple hypothesis that if a calcium channel would really exist in the mitochondrial membrane, it must be present in metazoa, including vertebrates and higher eukaryotes, and also be conserved to unicellular eukaryotes like trypanosoma, where mitochondrial calcium uptake was also recorded. But this channel should not be found in fungi, in cerevisiae. So if we translate this simple observation into an evolutionary genomic exercise, we will be actually searching among the mammalian mitochondrial proteins that have an homologue in metazoa, that have an homologue in protists, but they are not conserved in yeast. And this approach prioritizes only 58 genes out of the whole human proteome, including the MECU1 protein that is at the time was proposed to be an EFN protein regulating mitochondrial calcium uptake. 1 year later, also this was followed by the discovery from both the Muta lab and Rizzuto labs of the mitochondrial calcium uniporter that was later on proved to be the pore-forming subunit of this channel, and followed by the identification of many other components. So we now know this channel is actually a macromolecular complex. But what is interesting is that the majority of these components, except for EMRE, were identified also based on paralogous relationships, so on homology relationship to the founding member, to MCU and MECU1. So, however, as I said, EMRE was constituted an exception and didn't seem to co-evolve with the complex. And altogether, these studies advanced the field, but also pointed to several inconsistencies, especially in the evolutionary origin of MCU. So based on, let me go back. So based on what I've just shown you, you would expect that MCU and MECU1 would always co-evolve, meaning that they would be both present in organisms that show mitochondrial calcium uptake, and that fungi would not have any homologs of the uniporters, at least not of MCU and MICU1. However, an evolutionary analysis done by the Muta lab across 138 eukaryotes, compared to the 5 that were actually used to identify MCU and MICU1 initially, this show a very peculiar pattern of MCU and MICU1 distributions. As you can see here, many fungal species were found to have homologs of MCU in the absence of MICU1, so in the absence of its regulator. And based on these results, several speculations were made, including the following. The first is that fungal MCU were considered functionally homolog of the human MCU. That means that both MCU would act as a calcium component. The second is that fungal MCU would actually be sufficient for mitochondrial calcium uptake because they were the only component found in fungi. And more importantly, that MICU1, which co-evolved so well, so strongly with MCU, would be insensible for mitochondrial calcium uptake, at least in fungi, or that there would be another regulator that we were not aware. So 1 year later, the same group identified a protein, as I said, EMRE, by co-IP analysis with MCU. And EMRE was considered to be an animal-specific innovation of the uniporter. So a component that the group proved to be essential for mitochondrial calcium uptake in invertebrates, but that would be only in animals. So it would be a special component that the animal system invented in order to make MCU functional. And overall, all of these assumptions led then later to several independent structure studies that focus on dissecting the basic principle of uniporter channel assembly and the mechanisms of MCU-driven calcium uptake and selectivity. This despite the fact that we and others already back in the 70s showed that fungal species and fungal MCUs lack uniporter activity when, for example, you express them heterologously in HeLa, devoid of MCU component, or in yeast cells which do not have any calcium uniporter. So for all of these reasons, we made an effort to try to solve these paradoxical observations. And so we decided to perform a very careful inspection of the overall taxonomic distribution of the MCU components across as many sequence organisms as we could find. So in this case was over 1,000 eukaryotic genomes. And for the sake of time, I will simplify this picture, very complex, using a cartoon. So what you would see here is that based on our phylogenomic analysis, we hypothesized that the MCU-MICU complex emerged at the origin of eukaryotes. And therefore, those two proteins would represent the minimal core component of the eukaryotic uniporter channel. Both, as you see here, have been concomitantly lost many times during the evolution of eukaryotes, always co-lost or co-present. And in some cases, this loss is correlated to the presence of relict forms of mitochondria in these organisms like Giardia lambia. But contrary to previous efforts from the analysis on 138 eukaryotes by Muta, we find that, or at least our analysis suggests that there would have been one duplication event at the origin of the opisthoconch, which includes actually the, let's say, the holozoa, so the vertebrates, the animal, and the fungal clade. And this duplication would have led to the existence of two MCU gene family. One that we call animal specific, and that it was conserved holozoa all the way to animal, and the other that we call MCUP for fungal specific MCU that was actually conserved in fungi. What we found are fungi here before the split of the animal and the fungal lineages that actually had both paralogs, MCU and MCUP. And what was most striking is that we identify EMRE sequences in these organisms, suggesting that MCU, MIG1, and EMRE are the core component of the opisthokonт uniporter. So EMRE is not a metazoan invention. So this was, we decided to actually try to confirm experimentally this observation. So we look at those species where we find the presence of both MCU paralog, the animal specific and the fungal specific, as well as EMRE species. And then we use a synthetic biology approach to reconstitute mitochondrial calcium uptake in yeast. So here we express different combination of these components, MCU and MCUP, with their relative EMRE sequences. And we trigger intracellular calcium release by glucose-induced starvation, which in yeast is known to, in presence of extracellular calcium, to induce the entry of calcium in the cytosol. This is then taken up by the reconstituted uniporter, if functional. And we can record this calcium uptake by the presence of a constitutively expressed aquarin as a calcium sensor. And so what you see here in the graph is that only when we co-express the animal-specific MCU, which we find in these fungal species, with their relative EMRE, we can detect a very nice calcium uptake, which is similar to the one that we measure when we co-express the human MCU and the human EMRE. But this is not true when we use the MCUP as so the fungal specific MCU, suggested that these MCU homologs that were initially detected in Basidiomycota and the species that have been used for structure studies are not functional orthologs. They don't function as a calcium uniporter. Maybe they did, but they do not retain this function. And so we also took, so this study also allowed us to, now that we have a group of MCU functional sequence and non-functional sequences, allow us also to do several analyses of sequence diversity to try to understand what are the genetic determinants of these functional differences. And what I have no time to present this part of the work, but of course you are very welcome to read the publication that just came out months ago. What I want to show here is that, interesting, although human and fungal EMRE sequences are very divergent, so these are all, let's say, functional EMRE, and they're very divergent, the human MCU can work with all of them. In some cases, we found even like a better calcium uptake response with some of these fungal EMRE sequences. While the fungal one only can work with its own EMRE, suggesting that this C-terminal domain that you only find in the fungal species is important for the regulation of the uniporter function. And so that's, other than looking at structural similarities between fungal and animal MCU, like many of them the following structural studies of human MCU are doing, under the assumption of a functional homology, one, I would say, should rather look at structural differences, which may, in fact, highlight important residues or domain for MCU function. Another speculation that was made in this study is actually that we know everything about the uniporter complex. It was defined as the holocomplex, like the uniplex. we wanted to challenge this hypothesis, and the reason why we were skeptical about the possibility that the uniporter would be identified completely is that most of the studies that have tried to identify these components have rely on functional, so on one approach, mainly on functional genomics on one side, and on the other side, on biochemical approaches that used systems where the component have been overexpressed, like in these studies. And we know now that the overexpression of one or another component of the uniporter can change both the level and the stoichiometry of the complex. We then moved to develop proteomic and biochemical strategies where we try to look in three directions. In one case, we look at the identification of protein-protein interaction between uniporter component and other mitochondrial protein. We also look at the abundance, the absolute abundance of this component in the cell, and combining these two level of information, we can actually deduce also the stoichiometry of the complex in the cells. In this case, we focus on hex cells for simplicity. So let me introduce the biochemical approach. It's quite straightforward. What we use here is a hex cell line where we express one of, in each cell line, we express one of three structural components of the uniporter, the MCUB, MCU, and EMRE. Those are the membrane component of the channel, and we express them in only one copy, so a near endogenous expression in a specific locus using this flip T-Rex system. The tag is that we attach at the C terminal is a combination of a streptavidin and his tag, each HA we use for detection. And then based on tandem affinity purification done in several biological replicate, we compare the interactome, so the protein-protein interaction from the tagged MCU component with the eluate, so let's say of the interactome of a control cell line where none of this component is actually expressed. So this would be the parental cell line. And then we do unbiased mass spec analysis of the pull down samples. And we plot this data, we analyze this data as a volcanoplot. For who is not familiar with this type of representation, what you do here is you plot the enrichment, so the log2 fold change of the protein that you identify and evaluate by mass spec with the p-value. That gives you, therefore, if you see some protein here, it will tell you that this protein is not just enriched, so it's not just found as an interactor in the tagged in the tagged version, but compared to control, but also that this interactor is statistically significantly enriched. So as you see here, as we suspected, the expression of this component, a near endogenous condition, didn't alter the level of any other of the known component of the uniporter, except, and this is very nice to see, except for EMRE. So here the expression of EMRE was followed by a slight down regulation of the endogenous EMRE. So also corroborating the idea that the level of EMRE is very well balanced. Its homeostasis has to be tightly regulated. Then we confirm, of course, by pull down, Western blot, and also by proteomic that the bait that would be enriched in our eluate compared to whole cell lysate. And when we analyze this data, and here I only show the results for the MCU tandem affinity purification, but we see the same for the other two. Was nice to see that we could recover basically all of the known component of the uniporters. But as you can see, we also identify some additional one. And so what are those additional one? It's here, what you see here, it's a comprehensive view of the interactome of the MCU, of this, starting from these 3 baits. And you can interpret this network in 2 ways. On one side, we identify component, we think component, of course, that are strongly interacting with all 3 baits that we use, suggesting that these components may be actually new subunit of the uniporter. On the other side, you can also interpret this network as a way, as a tool to look for the molecular links between calcium uptake, so MCU-dependent calcium uptake, and several mitochondrial processes. We find components, proteins that are involved in several cell death pathways, not just apoptosis, but also ferroptosis and necrosis. We find kinases and phosphatases that are somehow associated to the complex. We also find, interestingly, several transporters of metabolites and that are calcium regulated. And so this would allow us to also discover new mitochondrial processes that we were not aware to be regulated by calcium. What we focused on as a proof of concept of this analysis was to follow up some of the candidates with a different, that were identified either as a specific interactor of one bait. In this case, is a protease of the MRE protein, which we show has an effect on calcium homeostasis in mitochondria. Well, we also follow up the MECU3, and I'll tell you in the next slide why. We made some effort to characterize this component, which is unknown, has an unknown function, and also to one protein that is involved in the cardiolipin pathway in mitochondria. And cardiolipin has been already shown to be important for the MCU assembly. So MECU3, we decided to confirm the finding of MECU3 as a part of the uniporter. This is not surprising, given that it was already published and mentioned that MECU3 is a regulator of the uniporter. What we found interesting is that we find MECU3 in HEC cells. So this finding, where there are also MECU1 and MECU2. So the presence of MECU3 is not mutually exclusive with the presence of MECU2. And it could actually perhaps explain some differences physiologically that people observe between HeLa and HEC mitochondrial calcium dynamics. So we are not the only one that found MECU3 in HEC cell. There were already some proteomic analysis done by the MaxQuant in different cell lines that detected the MECU3 in HEC293. What we show is that we confirm by lentiviral mediated knockdown that the reduction of MECU3 protein level in HEC has a consequence in the kinetic of calcium uptake. So we see a decreased calcium uptake, which fits with the data from that have been already published by Diego De Stefani and Maria Patron, where they show that the overexpression, and we recapitulate this finding as well, they show that the overexpression of MECU3 in HeLa cells, which do not have MECU3, lead to a faster calcium uptake, proposing that MECU3 is actually an activator of the uniporter. We then, going back to this completely uncharacterized protein, we also we were quite intrigued by this because our data suggests that this would be a novel subunit of the uniporter. So we did again lentiviral mediated knockdown, and we showed that when we eliminate or decrease the level of this protein by either SH RNA, and we also see the same by SI, so by stable and transient, knockdown, we see a dramatic increase of the level of calcium in the matrix. And in fact, the knockdown of these proteins is quite toxic for the cells. We do see this kind of phenotype independently of whether we trigger calcium uptake in the cell by the ER pathway, so by the IP3 receptor using histamine as a stimulus, or if we go through the CAC channel. And this is not a mere consequence of the fact that there is an increase in the cytosolic calcium level, so that the effect of the protein on the mitochondrial calcium uptake is not upstream. And this would be, let's say, obvious, given that we co-IP the protein with the MCU, and we demonstrate also that it's enriched in mitochondrial fraction. So it shows the mitochondrial localization. But the most, let's say, strong evidence is that when we go in permeabilized cells, where now we exclude the involvement of any other pathway upstream, we do still see the increase in mitochondrial calcium uptake, which is ruthenium 360 dependent. So to make a summary, I have no time to present you the other half of the interactome story. We do also look at, but I would be happy to discuss afterwards if you have specific questions. We do also look at how this interactome remodels in response to genetic perturbation and specifically to the knockdown of each uniporter component. And we also get insight on the stoichiometry, on the relative stoichiometry of these and how this changes in HEX cells. So to summarize, we think that this MCU interactome network would be a quite rich and helpful resource, we hope so, for all of you. To expand our understanding of how the uniporter functions and how mitochondria processes are linked to calcium uptake. And well, for the future, we are now starting several projects that now try to go from in vitro to in vivo. So the same question we asked with the MCU, trying to map the MCU interactome in HEX cell. We are now doing it in a tissue-specific fashion, generating mice that have several components tagged. We are also very engaged in mapping signal transduction cascades in the mitochondria that are mediated by calcium entry. And we have now new data on perhaps novel components that also mediate the mitochondrial and ER calcium signaling. And then, of course, some of these, when you predict new components, any of these components could be a valuable new disease gene and a valuable drug target. So that's also a direction we are moving on, and we plan to move on in the future. And with this, I would like to thank especially the people in my lab that have been involved in these studies, including the former members of the lab. And all our collaborators. One of these is Yuri, so I thank you again, not just to chair it, but also to collaborate with us. And I write here my email because we are always looking, of course, for a very motivated student, technician, and postdoc to apply to our lab. And with this, I would be very happy to answer any questions. Thank you very much for your listening.
Thanks, Fabiana. I don't know how to clap on this platform, so I just use my hands.
I will use the mode in case I need to go back and forth for answering the questions. So I will just leave now this slide.
So I am supposed to present to you the questions, and there are several good questions. The first ones are from Kevin Foskett. And Kevin asked to clarify whether the fungal MCU structures that were published in 3 Nature and 1 Science paper last year are structures for functional or non-functional calcium channels. Kevin also mentioned that as far as he remembers, some of those papers showed calcium transport studies. So could you please clarify this?
Kevin, nice to know that you are in the audience. Based on our analysis, the fungal species that have been analyzed by structural studies in these publications are non-functional homologs of MCU. That's what we call MCUP. And yes, you're right. In 2 out of these 4 papers, they show constitution of calcium uptake by expressing the fungal MCU homolog in bacteria. But interestingly, this paper, they actually mentioned, they don't show the data, but they mentioned that they attempted to reconstitute calcium uptake in yeast and human cell, and they couldn't see anything. And that's what we get, and that's what other people have got in the past. Based on on this analysis, those people have sequenced would be this fungal ortholog, and this we show by reconstituting them in HeLa MCU knockout cells, that they are not able to rescue an MCU, human MCU loss of function. This is part of the supplementary figures. Also, when you overexpress this one in wild-type HeLa, you don't get a gain of function. And when you express them in yeast, you cannot reconstitute calcium uptake. We think that this evidence would be enough to somehow corroborate our statement compared to a system, which is the bacterial one, that is not even a eukaryotic system, right? Also, what they have observed in this studies, I mean, they had to use quite a great amount of calcium to see some uptake, and the uptake kinetics didn't even get close to what we see physiologically, while we are using a system that it's in vivo. So in vivo, we can actually recapitulate an uptake, a rapid uptake. I hope this would clarify Kevin's point.
Yes, I think so. And he followed up with a very specific and interesting question, whether you see any evolutionary relationship between MCUP and MCUB.
So the MCUB, let's see if I have, we do not see this coevolution. So MCUB, let's see if I have, I'm afraid to get out of the central view, not to lose you, but yeah. So the MCUB is actually unrelated to MCUP, so the fungal specific one. And what you see here is that MCUB appeared later on during the animal evolution, so as an additional event of duplication of MCU. So it's not, while the MCUP and the MCU duplication occurs at the ancestor of this lineage. So we don't think that MCUP and MCUB are actually homologs. We think that MCUP is working, as I said, either it had this function initially and then it was lost, or has some function that we don't know about it yet.
So the rest of the questions about the second half of your talk about the proteomics, Roberto Docampo asks you about your TEP experiments, whether you pull down any ATP synthetase components like he pulled down in trypanosomes.
Oh, that's a good point. That don't come up out of my mind right now. We do see some, yes, we do see actually, we do see some subunit of the ATP synthase, and we also see some subunit of the complex 4. What you see here as orange. Now I don't remember exactly which one. But yeah, we do see some of these components.
So moving from the inner membrane to the outer membrane, Geert Bultink would like to know whether you have any mitochondrial outer membrane localized partner for MCU or the other components.
Yes, we do. Some of these are outer membrane localized. So, but on the so some the majority, those are very, very few, and we don't identify anything like VDAC, if that's what you are thinking of. We do identify specific mitochondrial proteins that are mainly in the IMS, so in the intermembrane space, and are known to be associated with the outer membrane. We also do not identify NCLX, which is a bit surprising, as we would expect it being the exchanger to come down. And I want to clarify, we do not see in our reciprocal, we have another study following up on MCUR1. We do not see any MCUR1 in our pull down if we use any of these feed baits or if we use MCUR1.
Okay, could you please tell anything about MCUR1? Is it coming down in your...
No. As I said, no, it's not. We don't see any MCUR1 associated with the pull down. If we use EMRE as a bait or MCU or MCUB, and we don't see any MCUC component when we use MCUR1 as a bait. So we have another study where we try to characterize, again, combining several approaches, so what could be the role of MCUR1, as we think it has a much more fundamental role, and its dysfunction would lead to a calcium phenotype because it's actually disrupting mitochondrial function heavily. We identify a patient with an MCUR1 dysfunction, and yeah, we think that this protein is involved in amino acid metabolism pathway.
By the way, this was a question by Jonathan Sobolov. Clearly, Philadelphia is very active in the conversation. Another question from Kevin Foskett. What is MCNR1? You mentioned this protein, but can you say more about MCNR1?
Okay. So it's as I said, it's a completely unknown. It's a protein of unknown function that has been. So we don't know anything about it, except that it has been associated with some inflammation processes. There is not much really to say other than we do find it very interesting because. Of course, it's uncharacterized and it's strongly pulled down by all three baits. We have we do look at the localization, and we think that the protein is localized in the intermembrane space. We also we are now trying to we have some evidence that this protein could regulate mitochondrial calcium uptake by regulating the assembly of MCUB within the complex. And so, in fact, it mimics the MCUB phenotype. If you knock down MCUB, you have an increase of calcium We also see, but at the moment we are still studying. As we only know that it's an unknown protein with an unknown function that we strongly identify as a component of the complex, and we do see a very striking effect on mitochondrial calcium uptake, which is mitochondrial dependent, because we do see impermeabilized cells, so isolated mitochondria. Unfortunately, there are not ways to follow it up. Being unknown, there are no good antibodies, so some of these analyses are hampered by the lack of good reagents.
So let me ask you more on this. Do you see a phenotype in the MCUB knockout? You mentioned that it may act through the interaction of MCUB with the complex. So have you tested MCNR1 in the MCUB knockout cells?
Yes. So we have done. We have looked at knocking down the MCNR1 in both transiently knocked down MCUB HeLa cells, as well as stable MCUB knockout HeLa cells. And they show the same phenotype. So it's not additive. As if the absence of one is already somehow leading to the lack of effect of the other. That's what you were asking? Or?
Yeah.
Okay.
So a question from Antonio Villalobos. Tumor cell mitochondria have a great capacity to accumulate calcium. Anything known about the subunit composition of the uniporter in tumor cells versus normal cells?
We haven't really looked into this. So, I mean, it would be possible to, let's say, compare systematically this interactome in different cell type. The only thing that may get close to a kind of an answer would be perhaps this. So here we show the tissue specific expression of all of the components that we pulled down by the 3 bases, and some of these components have been already involved, are already annotated in OMIM as cancer genes, but we haven't looked into that systematically.
Okay, you mentioned that your new proteins might allow mitochondrial calcium to affect more functions, new functions of the mitochondria. So Natasha Prevarskaya asks you, what are the direct targets of calcium in mitochondria to assure specific physiological cell functions?
So the direct target, I'm not sure I understand the question. So you mean that whether we can actually imply from this analysis that these components would be direct target of calcium, or?
Well, that's the way I would take the question as well. Okay. Natasha, if you want to clarify, please just drop another note. So could you please answer this way?
So what I can say is that this is a pull down experiment, right? So of course, and you cannot expect that all of the proteins that co-IP with the bases will be part of the complex, right? Otherwise it would be the whole mitochondria almost. So what I meant to say at the beginning is that you can see this map in 2 ways. on one way, it could contain novel component, so novel subunit of the channel, like we think this is the case for MC-NR1. And on the other side, it could point to functions, like, for example, we find proteins, mitochondrial proteins involved in transcription and translational regulation to be statistically significantly enriched in the MCU-CL weight. This could suggest that calcium uptake in mitochondria through MCU is coupled or is regulating pathway like transcription or translation, is regulating ferroptosis, which already there are evidence, is regulating lipid metabolisms, so that the implication would be more a functional link than a physical interaction.
Okay, so Shaishin Shu asks you about your opinion on the lack of functional phenotype in MCU knockouts, like the Toren-Finkel heart or the more recent fat cells by the Muta group, which describe lack of functional change in the MCU knockouts.
This is a very broad question. I would say that as many have already discussed this point in several reviews, this could be on one side the consequence of the specific genetic system that was used by the Finkel group, as other, as we know, the knockout of MCU in Black 6 is lethal. The lack of phenotype from that sometimes is observed in the absence of MCU could also be a consequence of some adaptation event. I think this play a strong role, as even the Finkel group show for the MICU1 and the EMRE. Some people speculate about possible other entryways, but in absence of MCU, as far as I know, there hasn't been any detection of calcium in the mitochondria. So that would be my opinion, my, let's say, comments to the question.
Well, let me add here that some of those papers claim that there is some calcium inside the mitochondria. They don't specify a transport pathway, but the Finkel-Murphy group and some other groups showed some evidence that there was calcium inside the mitochondria in the MCU-KO cells. So they propose that there should be some calcium transport system, although we don't know about dynamic activity of such a pathway. So we also have another question following up on Geert Bulting's question. Any of the OMM proteins in the interactome have a sizable cytosolic domain? And this is a question by Yuri Chordash.
Let me see. So I am embarrassed to say that I honestly do not remember which one of those proteins, but I would be very happy to answer the question if they send me an email. So I'm sorry, but I don't recall now the specific proteins in the network that are outer membrane localized. But as I said, if you drop me an email, I will definitely be happy to answer.
Yeah, this is something that I wanted to mention in my introduction, that over the years, I noticed how open you are in the discussions, and I really encourage people to contact you because you are a tremendous source of information, and I think great potential collaborator. So let me also ask one question. There is this growing line from Lucas Corano, Wolfgang Greyer, and John Erard talking about interactions of the uniporter with Mycos complex of the Christie opening. And they propose somewhat different models for the interaction, but all of them imply some Mycos complex components. So do you have any clarity from your proteomics that you can share with us on? Okay.
This component, this protein is the IMMT, so it's one of the Mycos component. I don't remember if we identify others, but we do see an interaction with some component of the Mycos.
Both MCU and MICU?
So both. Okay. Well, we do see this only if we from the EMRE and MCUB side. We do not detect this component in the if we pull from MCU, but this doesn't mean that it's. I mean, a negative result in proteomics. It's not a negative. It can also be not a negative result. You know, it can be just that we didn't detect or it wasn't enriched enough. So I can only tell you that we do see an interaction if we pull from MCUB or EMRE side from this.
Okay, thanks, Fabiana. If you look at the chat, it is pegged now with congratulation notes. Okay. So I don't read those. You can read those by yourself. And if there are no more questions, I mean,
to add a comment that. You know, as you said, many of the studies that we do are more focused on developing and applying an approach to like advance the understanding, and most of the times we are faced with like in this case with more hypotheses than what we can test. So what we really hope our contribution would be to the field is that, you know, we could generate useful resources for other people. So as you said, I'm always very happy if someone drops me an email and is willing to collaborate, and definitely most of the times very happy to share. Thank you very much for the congratulations. I enjoy, as I say, it's strange to give a talk in front of a ghost audience, but it's nice to know that all of these colleagues are in the background.
Thanks. So we close the big platform, but feel free to stay connected. If you want to chat with Fabiana, she will be available